Drivers once again. Wow, I'm looking like the Snapchat filter with these blue light glasses, but welcome back. Today we're going to wrap up chapter 22 by talking about how this all comes together. So all of the things that we've discussed in chapter 22, how does that result in speciation? So there's a lot of questions that many people still have about how long it really takes for a new species to form, especially because we consider evolution usually to happen over a long span of time. And we also don't necessarily fully understand how many genes are needed to differ in order to have a new species, in order to be considered a new species. We do a lot of studying of different things to help us understand how new species arrive. So we could study the fossil record and study what we can find on Earth. Morphological data like their physical characteristics and the way their body is organized, and also molecular data, so looking down at the level of the genes, the chromosomes, and the amino acids that are produced in transcription and translation that cause certain genes to be turned on and off. The fossil record shows us a lot of patterns, but we, interestingly enough, see a lot of sudden species that arise in the fossil record. And sometimes those species will show up in the fossils, they will not change, and then disappear all of a sudden. And so scientists call this period where we have just a kind of a static, regular record all of a sudden with a sudden change. We call this punctuated equilibria. So we have periods where it's kind of there's an equilibrium, not a lot of change, and then it's punctuated by change. And this contrasts with a model of gradual change in species existence. So, for example, we have these two fish that look very different, obviously, but we've observed a hybrid of those two fish that's been a result. And also, what this punctuated equilibrium in the fossil record indicates to us is that we might actually see that species can arise a lot faster than we thought. We could get a new species very quickly. So a lot of this section is going to focus on this sunflower, Helianthus anomalus, which was the hybrid of two other sunflower species that quickly became a new species. So the punctuated model, we maybe have one species that becomes all of a sudden diverges into two species, and it happens very quickly, compared to the gradual model where we have one species and then we see these slow changes that over many, many, many years cause a new species. And it's not to say both of these don't exist, but for a long time we thought only the gradual model was possible. But in the fossil record, we see this punctuated equilibrium. So here's that sunflower we were talking about. We see new species arise every 4,000 years, in some cases, to 40 million years between a new species forming. But the average between a new species forming in a particular organism or population is an average of about 6.5 million years. So then the question also becomes how many genes are required to change for us to have a new species? And that answer to that so far has really been it depends on the species that we're talking about. So there's some species where an entire new species has arisen due to the change of only a single gene. But other species, we don't see a new one arise until we see a change in many genes. So for example, there's these Japanese Euhadra snails, and the direction of their shell spiral is controlled by one single gene. But if you recall from the reproductive isolation, if their shells spiral in opposite directions, there's a mechanical issue and they cannot reproduce in that way. And so that affects their mating ability. So we see that there. In monkey flowers, there's two genes that affect flower color, and that influences what pollinators are going to come and pollinate those flowers. And so the monkey flowers with alleles at the flower color locus, if they have different alleles there, they're not going to interbreed because they attract different pollinators. Some may attract birds, some may attract bees, etc. And then here you can see this is the sunflower genes. So we can see here's these two different gametes, two different parents, and this sunflower was a hybrid of those two parents. And you can see the experimental hybrid compared to the H. anomalus, which was the hybrid they see in nature. We can see on chromosome 1, they're exactly the same genetically, but on chromosome 2, we have just a few differences here, here, and here. So for many other organisms, speciation can be influenced by large numbers of genes and gene interactions, like we see with the Helianthus sunflower. There is about 26 chromosome differences that cause that species, that hybrid species, to become its own species. So essentially, when we're looking at macroevolution, we started off in the evolution unit talking about the very, very small scale evolutionary changes, the changes in the allele frequencies. But as those allele frequencies accumulate, we eventually see large scale evolutionary change, which results from the cumulative effect of many species arising and many species going through extinction. And over time, we see those big sweeping changes, which we consider to be macroevolution. So that's really the end of this section. I hope you enjoyed learning about how new species are formed, and we'll see you guys next time. Looking forward to reuniting with you subscribers soon.